So we talked about this before in the context of SLIM and in the context of Gibson cloning and sewing. The problem with restriction enzyme cloning is it has severe limitations. So one of the limitations is size of things. The bigger they are, the more likely it is that they're going to have internal restriction enzyme sites. So it's sequence limited. So it's limited by sequence. Also, if you do, now let's talk about the limitations. I should say, the reason I introduced SLIM and Gibson and sewing was because I used it as a way to address those concerns, the limitations of restriction enzyme cloning. But now let's talk about some of the limitations of these PCR-based methods, because these also have limitations. So SLIM, Gibson, all those things involve making primers. And in your primers, you usually have regions of overlap, of homology. So if you amplify, say, gene X, this is an example of a sewing reaction. And you want to fuse it to Y. I'm actually going to erase that. You can make primers that match. And then when you amplify these sequences, you can get amplicons that can prime each other. And that way, when the polymerase in the PCR reaction then continues to fuse, it fuses them together. So that's an example of like a sewing reaction. But what would be a limitation of this process? Can you guys think of any?
It could be they don't line up exactly.
Let's think about what goes into the reaction. So what would go into this reaction? It's a PCR reaction. So what goes in there? Primers. What? Buffer. DNTPs. Water. Polymerase. Okay. So think about limitations of the polymerase and think about limitations of the primers. So in terms of the primers, we talked about what happens when you start to make really, really long primer sets. You start to get hairpins, and hairpins will kill your reaction. So there's a limitation in the sense that when you do these reactions, you're making your primers longer and longer and longer. And some of these primers need 40 or more base pair arms. Okay, what's the limitation of the polymerase? It can only go so fast. I don't know if I'd say, I mean, it would, but I think the bigger concern is again, the mutation rate. It's the mutation rate. So even the best polymerases are 1 in 10,000. What if you need to amplify something that's 30,000 base pairs, or even 50,000, or even 100,000? It's not even possible. It's not possible to do that with fidelity. So there's a limitation. If you need to clone big stuff, you just can't do it with a polymerase. And it starts to get really difficult greater than 5,000 base pairs. Okay? So there are ways that we can clone things that deal with these problems. One of these strategies is called Recombineering. Okay, so what do you think Recombineering involves? Recombination, right. So homologous recombination, if I just write HR, that's homologous recombination. What is homologous recombination? I think this is review. I think I've talked about this before. So homologous recombination is actually, it's a form of DNA repair. It's a form of DNA repair. And it's also, in sexual organisms, it's a form of mixing DNA, okay? But in things like bacteria, which is mostly, or yeast, which is mostly where we're doing recombineering, we like to think of it in a sense of it's a repair mechanism. So what happens in a cell? If you have a chromosome, and it breaks. So let's say you get a cut right there. So this would be called a double strand break. So if you see DSB, double strand break means if you have a sequence of DNA, double strand break is when you literally cut both strands. Does that make sense? So you can have a single strand break. Single strand break is a lot easier to repair because you have, if you just have a single strand break, let's say like that, it's very easy to repair because something can come in and just read off this template right here and just refill it and then ligate it, right? That's pretty easy. If you have a double-stranded break that looks like this, it's a lot harder to repair because the cell sees this and it's like, I don't really know like what goes in there. Like, am I missing something or what? Okay. So there's two ways, or well, there's more, but in general, there's two ways to repair double-strand breaks. Okay, and this is actually, this will come up again when we talk about CRISPR, because CRISPR is actually really, it's a nuclease that induces a double-stranded break, but then when you edit the genome, it's actually all entirely based on the double-strand break repair systems. So one of these is basically called blunt end repair, and one of these is called HDR, or homology-directed repair. Or in other words, you could say it's homologous recombination. It's the same thing. So blunt end repair is when the cell just takes the two pieces of DNA and it just literally smashes them together. So you've taken those two things and you just smash them together, and then a ligase will come and fuse the backbone. Okay, so that's blunt end repair. But like I said, the cell, that's kind of, this is usually what happens, blunt end repair. But it's not the best way to repair a double strand break because you might have lost some sequence. Does that make sense? If you just smash things together, you might have lost something. So homology-directed repair is the other way to repair double strand breaks. And homology-directed repair is if you have a double-stranded break, the cell's going to look around for something that matches. And if there is a matching piece of DNA, so this is a case where this matches this and this matches this, the cell can then recombine these regions and basically fill in what was missing from here. And it replicates this, okay? So you can get perfect repair based on homologous recombination.
So is that other piece of DNA that's intact that it's using to repair? That's like you have two alleles for every gene. Right?
Yeah, so it's a lot easier in diploids, right? Just like you're saying, because diploids are always going to have a second set that they can repair from, right? But how would a bacteria do it? Well, I told you in some cases E. coli is actually replicating for its grandmother cells. So there are cases where E. coli might be aneuploid, where it's got multiple copies of its chromosome floating around. Also, imagine if you're on a plasmid. Most of the recombineering we do is on plasmids. So say you have a high copy origin, which is 600 copies per cell. It's very, very easy to find a template for homologous recombination, right? So homologous recombination is what we use, is another means of cloning things, and it's independent of restriction enzyme sites, and there's no mutations because the polymerase is not adding in A, T, G, C. Does that make sense? It's just taking something that's already there and then like switching it out. Okay, so let's talk about how we use this. Okay, so I'll also introduce. Yep.
So in the
Homology-directed repair.
So you said that it doesn't use like DNTPs.
Well, it does. I guess that was a bad way to say it. Because
Just like
because
takes it out, and the other ones.
right, it does. That's a good point. So there's a strand invasion, and then it uses the template. It uses the other piece as a template to like read and write. But that's a good point. And a polymerase would do that. So I'm trying to think of why the mutation is less, why the mutational risk is less? That's a really good question. I have to write that down. But I know that the mutational risk is less.
I guess it's like you said, you can open book quiz.
It might be, but even in PCR, I mean, even in PCR, it's reading from a template, right? So I can't think of the answer to that right now. Can you send me an email right now? Write that down, and I'll look at that. I don't know the answer to that question, but I should. Okay, so let's move on to talk about how we do this. So there's machinery that controls homologous recombination. Okay, and by machinery I mean like there's enzymes. There's enzymes that do this. Okay, and the ones that we use are from bacteriophage. So I've talked about lambda before. I've talked about the lambda phage as a vector for cloning. Okay, and lambda, I told you, is a bacteriophage. What's a bacteriophage? Right, it's a virus that targets bacteria. Okay, so that's a bacteriophage. Let's talk about its life cycle. We've talked a little bit about it, but let's do it again. So there's two key points, lytic and lysogenic. I'm spelling it wrong. Okay, what's the lysogenic phase?
Is that where it infects? Oh, lysogen.
It bursts the cells. And then the lytic phase is the one where it integrates. Somebody double check me on this. I'm pretty sure. But that's the concept, is that there's one phase where the genome is, so if this is a chromosome of bacteria, there's one phase, life cycle phase, where the phage genome is just sitting in the chromosome. And it's just parasitizing the bacteria, because every time the bacteria replicates this chromosome, it also replicates the phage DNA. Okay? And it's content to be there unless conditions get bad. So if stuff gets bad, the virus wants to get out. It wants to, it's just like if you're on an island that's dying, you want to get somewhere else, right? You want to get out. So they will induce, the virus will burst the cells by stereotypical virus activity. So it'll make particles. That means that its DNA will actually now be transcribed. So that means it'll make messenger RNA. That messenger RNA will make proteins. And then those proteins will make phage particles, which are little machines. And then eventually they make so many particles that the cell bursts and the particles spill out. And then they can go to find a new island. Does that make sense? So there's these two life cycles, lytic versus lysogenic. So let me ask a question. How does lambda know? How does it know when it wants to be in the chromosome or when it wants to make particles? Can you guys think of how it would sense that? Yeah, so so it does, but let's not go quite there yet. How the first step I told you is when the virus when the virus genome is integrated into the genome of the bacteria, what's the first thing that needs to happen for it to get to this life cycle to make these particles? What's the first thing that has to happen? What'd you say? Needs to be methylated? No, not methylation. I see what you're saying. The bacteria would have restriction enzyme methylase systems to protect itself. But what I'm looking for is the genes. So the genes of the virus need to be transcribed. Okay. So this is why I taught you guys fundamentally about bacterial genetic regulation, right? So what would control a transcription of genes? Transcription factors, okay? And you said heat, right? So the genome of the virus, so let's say this is the genome, the lambda phage is basically like a big operon. So it's a bunch of genes strung together. And there's a regulatory, remember how when we've discussed regulatory regions, there's often like a piece outside that encodes the regulator, which is under a different regulation than the actual genes that would encode the operon or the virus, right? We kind of remember that from like the air operon, the trip operon, the lac operon. This is the same concept with viruses, is that in this case, they have a regulator that's sensitive to heat. And when it's cold, the regulator is shutting off the virus particle production genes. So let's actually look at the map. If you read the paper, here's a map of these genes. I mean, each of these is telling you these are the names of these genes: EA22, hint, exo, beta, gamma, kill, CL01. These are all genes of the phage that do something. And here is the regulator. This is the repressor, CL857. Okay, and this is telling you PR. PR has its own promoter. Okay, PR. It has its own promoter, and it's inactive at 42. That's heat shock. Yeah. So what this is saying, what my notes are saying, is that at 42, so this is a heat shock. That's hot for bacteria. At 42, this thing, the repressor, stops getting made. Okay, and then what's going to happen to these? These things are going to get turned on. Does that make sense? So this is how, if you heat shock bacteria, the virus, the lambda virus, the phage, knows to start turning on its genes. So this is the regulatory circuit of the phage. Okay. So lambda doesn't like it hot. It wants to get out before things start boiling. Is basically the premise. We looked at the structure of lambda. Oh, there's other things I want to note. What's this? And this? Where have we seen those before? What have I talked about?
Oh, ATTB and ATTB.
Yeah, I told you that before when we were talking about integration in Drosophila. There was a 5C31 integrase, which was a virus enzyme. And I told you that enzyme's function was to take ATTP and ATTB sites and recombine them. And this is how a phage, if the phage genome has an ATTB site and it finds an ATTP site in a chromosome, it can integrate. Does that make sense? So what you're looking at here is in the virus, when it integrates, so what's going to happen is it's basically going to do this, and the ATTP will be here, and ATTB, it'll actually be half and half when it integrates. But it basically is able to take a plasmid and then do this with it. Okay? Now when it pops out of the genome, what do you think happens? There's a recombination here, and now it's circularized. It's genome. Does that make sense? And then this is what actually gets packaged into the virus particle. Okay? So what you're looking at here is common in virus biology, is these ATT sites, and the function of the ATT sites is to be the sites of recombination, where that's what allows both the genome to integrate and also to circularize and pop out. Okay, so I wanted to point those out to you because you'll see those in phages. I pointed out the repressor. I pointed out the promoter. Okay, let's talk about exo, beta, gamma. Okay, these are the three genes. So if we're talking about lambda now, this is the lambda genome. It's got exo, beta, gamma. These are the three genes that are essential for recombination of the virus. Okay, so if you have these things being made, the virus can integrate or pop out. Okay, it will recombine. So let's talk about what those things do. These are each, so these are the names of the genes, the ORFs, and they encode for proteins, which are obviously called exo, beta, gamma proteins. Okay, these are all enzymes. So exo is an exonuclease, and it degrades inward from 5' end, creating sticky ends. Okay, so what that means is, if this is, we can talk about this in two ways. DNA can be circularized or linear. So if it's an exonuclease, that means it has to come in on an end. It can only degrade inward from an end. And if it's degrading the 5' end, what it will do is, what's saying is it will chew up a little bit of the 5' end to create something that will look for itself. Okay, so you can imagine how if exonuclease is being expressed, it might promote conditions where this thing can then circularize, right? Okay, so that's what the exonuclease does. So by having sticky ends, you promote things that start to look for things to recombine with. What does beta do? Binds and stabilizes the ends of the DNA, the ends of linear DNA. So linear DNA in bacteria is usually not stable. How could that be? So imagine you're a bacteria. This is bacterial cell. And you have a linear molecule of DNA and a circular molecule of DNA. Why is it that a linear molecule could be unstable and a plasmid or a circularized chromosome could be stable? Why do you think that could be?
Well, on the linear DNA, it's just not closed. The ends that can be invaded or something.
Yeah, so your ends are exposed, and what does that expose you to? What did you just learn that having your ends exposed exposes you to? Exonucleases. So in bacteria, there's proteins being made that are exonucleases, and in general, if you put linear DNA in a bacteria, it will just chew it up. Okay, so beta stabilizes linear DNA in the bacterial cytoplasm. So beta is this protein that will kind of like hold on to the ends and protect it from further exonuclease cleavage. Okay. I think it's similar. So if you have the poly-A-tail, it would be 5, 3, and then I know there's something that progressively like eats up where they fall off or something like that. So yeah, it's I guess a similar kind of conceptualization. But I think the purpose is different. The purpose of the degrading messenger RNA and stacking it with the poly-A-tail is to kind of regulate the timing of transcription, to kind of have things slowly turn off. Whereas in bacteria, most of the exonucleases being expressed in the bacterial cytoplasm, I think are kind of like defense mechanisms. So if it ever encounters like DNA that it doesn't want, like some kind of a virus or something, it has ways to degrade it. What?
Do viruses go around the circular DNA, or?
It's mixed. It depends on what kind of virus you are. But most viruses, let's see, flaviviruses are circularized genomes. The laminophage is circularized. There are viruses that have like linear sequence. There's also, it doesn't even have to be DNA. Like there's RNA viruses. Flaviviruses are RNA viruses, but that's circularized. So viruses is really complicated. There's all kinds of stuff, different genomic structures of viruses. Okay, gamma. What's the last one? Gamma also inhibits nucleases. Okay. So basically the gist is that if you don't express these three proteins, recombination doesn't work. The phage has evolved these three proteins to allow it to recombine. Okay? So what we've done in recombineering is we have taken the beta, exo, gamma genes, stolen them from the virus, and well, you could do it in multiple ways, but let's just tell you the one that I'm comfortable with. We've put those on a plasmid under the heat shock, the heat shock repressor. So that means if you heat shock it, it turns on and the repressor leaves. So the repressor leaves if there's heat shock. And we put this plasmid, so let's say there's an origin, let's say there's a cassette, I think the cassette is chloramphenicol. We've put this into a bacteria, and we can induce conditions where the bacteria will want to recombine stuff all the time if we heat shock it. Because if we heat shock it, these proteins will get made, and then the bacteria is looking for stuff to recombine. So what we can do is we take a bacteria. This is the concept. We take a bacteria. Let's say we have a plasmid. So we want, this is plasmid A. And it's the same concept. We want to insert gene X right here. Okay? So what we do is we make a linear product that has homology regions that match those red sites. Okay? And then we give this bacteria both of these things by transformation. And then we heat shock it. So 1, transformation, 2, heat shock. That's going to turn on beta, exo, gamma. And it's a short heat shock. And then what's going to happen is as soon as those proteins get turned on, this that we gave it is going to start looking for this, and this is going to start looking for this, and then it will. I drew it bad. I should have drawn it up here like this. This is going to recombine, and gene X will now insert into our plasmid. So here's why. Okay, I've discovered the answer to your question in thinking this through. Here's why it's not subject to mutations. Because the only piece that's getting repaired is this tiny homology arm. Does that make sense? This part is not getting touched. So our entire gene X orf, there's no change in that sequence at all. That's just getting recombined in. The only piece that gets repaired or switched out is this thing right here. Does that make sense? Yeah, that's correct. Okay. Oh, this is complicated. Okay. So this is common. Let's talk about what we use this for. Useful for knockouts. Let's talk about how you would do a knockout. So let's say you got the bacterial chromosome and you got gene Y, and you want to make Y delta. You want to make knockout Y. All you need to do, pick 50 base pair homology region here, 50 base pair homology region here. You amplify. You could make this with PCR. Let's say you do what's CAN? When you see CAN, what's CAN? Kanamycin resistance cassette. Kanamycin is an antibiotic, right? Good. Or AMP, we could do AMP. Let's say this is an ORF, right? I'm talking about inserting a gene that encodes an ampicillin resistance cassette. Okay, so if we make this and we want to make a knockout of gene Y, all we got to do is transform this into the cell. So 1, transformation, 2, heat shock. And then there's going to be a recombination of that. Let's draw it out here. There's going to be recombination. And now the new chromosome is going to look like this. And this gene Y has popped out. And this thing will get degraded and lost. And this thing will be stable. And we can select for this now by making media that has what? Ampicillin. Okay, so then we've just selected for delta Y, knockout of gene Y. Does that make sense? So this is how you could use this. Let's talk about how. So it's useful for knockouts. I talked about how you could a standard way to do this is you could PCR. What you could do is you would design. Here's how you would design your primers. You would build your primers to match the AMP resistance cassette ORF. You would want to encode the promoter. And then you would pick homology arms on your primers that match the site, so it's site specific, that you want to recombine on from the bacterial chromosome, from back chromosome. This would be your primer. And then you would do a PCR reaction to amplify this. And that's how you would produce this thing right here. You could do that with PCR. But you don't have to use PCR. Imagine you wanted to recover. Imagine you wanted to recover a sequence of, say, bacteria X. So there's some sequence in bacteria X that you want to study. You don't have to PCR it. You could make a plasmid. Make plasmid. So let's draw these two components out. Let's say this is a big, huge bacterial chromosome. And I want to study this green region of DNA. Okay? We can devise a strategy through recombination. Let's say this is a massive thing. Let's say this is 20,000 base pairs from here to here. And we want to study this massive region of DNA. Initially, from what you guys knew, all you would be able to think of is, well, you'd have to do a primer here, and maybe if you get lucky, you could amplify that and clone it out by restriction enzyme. That's impossible because in that 20,000 base pairs, there's going to be restriction enzyme sites. It's going to be impossible to amplify. It'd be impossible to recover the sequence into a plasmid. So what we could do is we could build a little tiny reagent, plasmid A, and let's say we just clone, we spend time, so step 1 is clone in homology arms. Okay, so let's pick a tiny, tiny red spot here, tiny, tiny red spot here. So let's say this is, the red spot is equal to 50 base pairs. I should make 2 different colors. Oh, I'll just draw over that. So let's say we clone in 50 base pairs here at the multi-cloning site. We could do this with a synthesis or a Gibson, whatever. We create a plasmid that has a site that matches here and a site that matches here, right there. Okay, then all we need to do is extract genomic DNA from bacteria X. Okay, so we've extracted that, and then we transform that genomic DNA plus plasmid A into a recombination bacteria, heat shock it, and then this is going to look for that, recombine, and this is going to look for that and recombine. And now this thing is going to have this huge plasmid A, with this 20,000 kb sequence insert, and we did not use PCR in a sense that we didn't use PCR to amplify this huge 20,000 kb region. We just used PCR maybe to synthesize the original tiny homology arms. Does that make sense? So this is a way independent of PCR that you could clone in massive sequences. And we don't have to worry about inducing any mutations in this massive sequence because the only thing that's getting touched are these tiny homology arms by the exo beta gamma recombination system and RecA. RecA is a recombinase. RecA is an enzyme that will induce those holiday junctions. Again, watch the movies. The movies are good here. Okay, let's see. How much time do we have? A good amount of time. You make them, you pick them. So what I'm saying is, if you say, so this is a good example from what Dr. Wayne talked about last time. She when she was talking about how she went to advisor and she was basically like, well, I don't know what the sequence of DNA does. And the guy was like, well, study it. So maybe there's a sequence of DNA in some organism and you want to know what it does. So you want to recover that. This is a way, the only thing you need to know is you just look at the genome and pick little regions. So if you pick little regions that flank on the 5' 3' end of the sequence you want to study, and then you could clone those into your plasmid A, and then through recombineering, you could recover that sequence. Does that make sense? Let's look at the paper. So I definitely want you guys to look over this paper. Let's look at the figures, because this is the first paper where this technology was developed. So here's what I drew out. Here's what I draw. And if you read the paper, this would become more clear than the way that I've explained it. I've kind of failed and botched, but I'm doing my best. And here they're showing you exactly what I told you is they make a little primer. And these little blocks are the homology regions that they picked. And cassette, whenever you see cassette, that's like an antibiotic cassette. Okay. And so the target, that was like gene Y. They're showing you how to do a knockout. So they're knocking out a gene Y target in the cell. They just put the homology arms that match that target. And then what happens is in product C here, you get a recombination event. And what's inserted is the cassette and pops out your Y and your Y is lost. Okay. So that's how it's showing you here how you would make a deletion. What else is worth looking at here? Table 2. Where's table? Okay, so let's talk about CFUs. So in this paper, what they do is they develop the system for how this works. Okay, they build the plasmids that express the exo beta gamma, and they start testing it. And one of the things you always want to test is efficiency, right? Because you might be able to demonstrate that something works, but if it only works once in a million years, it's useless. So you want to demonstrate how, there's a big question if you develop a new method, how efficient is it? Okay. So they measure this with what's called CFUs. What's a CFU?
It's just colony forming unit.
Yeah, very, very good. Very good. So imagine you could measure the efficiency of a transformation. So let's say you have a bunch of bacteria in a tube, okay? And you transform plasmid A, and plasmid A has kanamycin resistance cassette, and you want to test the efficiency of your transformation. That means you're asking how many bacteria picked up my kanamycin plasmid. All you need to do is transform that and then plate that on a cell or plate that on a media with kanamycin, and you'll get your colonies. And you can quantify how many colony forming units there are by counting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 in CFUs. And you would do this per microliter or something. You know, you could quantify this. Does that make sense? Very, very, very simple concept. And now imagine you're testing different techniques. You're testing different cloning techniques. So imagine if one cloning technique gives you 11 CFUs per microliter, and another one gives you 500 CFUs per microliter. Which one is going to be more efficient? Yeah, it's very, very easy, very, very simple. So this paper is actually very, very, very easy to understand. So here they're showing you CMR, that's chloramphenicol resistance cassette. So it's chloramphenicol resistant recombinants. So they're measuring how many times they successfully recombined this chloramphenicol cassette into the chromosome. And what do you see? So let's look at, here they're telling you, what are they telling you here? Yeah, how long they heat shock. So if you don't heat shock, you get no colonies, right? You get no recombinants. If you don't heat shock, you get no colonies. If you do heat shock for 15 minutes, you get 2.5 times 10 to the fourth recombinants in colony forming units. So what they're proving to you here is, look, we built this system under a heat shock promoter, and they've just proven to you that the regulatory system that they built works. Okay, so they're telling you if you heat shock, recombination happens. Okay, so that's what they've proven to you in table 2.
And so the target sites are the different promoters.
Yeah, they've tested it in two different target sites. Yeah, so here they knocked out GalK, and here they're knocking out CLL kill gamma. That's actually the region from another region from the phage. So they're just kind of like two independent, like, let's do this twice.
And the bottom one works better.
That's correct. Okay, very good. Let's look at these. Let's look at figure 3 and figure 4. Okay, so here, recombinants. What are they measuring? CFUs, colony forming units. And look, they're just telling you, look, there's 1,000, 2,000, 3,000. Okay, and they want to know. Well, they're just laying out the protocol here for you. This is the first paper where they figured out how to do this. So they're literally telling you induction, which is heat shock, like how many minutes is it most efficient for? If you heat shock for 0 minutes, there's no recombinants. If you heat shock for 30 minutes, you kill the cells. If you heat shock for 15 minutes, you're right in this perfect optimization zone where you get the most recombinants. Does that make sense? Very, very, very simple graph. They're just mapping out how to do it. Okay, let's look at figure 4. So linear DNA. So this is what I was telling you. If you have bacteria and you want to recombine something in, you'll build that. What they're doing is they're amplifying a linear cassette. So they're using what they showed you up here. This is a linear cassette. Okay. And they want to know, well, how much of that should I put in? Like, should I put in like a whole bunch or just a tiny bit? Like, how much should I put in? If you put in 0, doesn't work. If you put in this many nanograms, so that's a measurement of DNA concentration, it's the most optimal, okay? You get the most recombinants. So you want to put like 300 nanograms of DNA of your cassette or of your plasmid, whatever you're going to recombine in, into the cell. Let's see, what are the other figures? Figure 5. Okay, so I told you that all you need is beta, exo, gamma. That's all you need to get this recombination system to work. But there is another protein, which I told you about, which is called RecA, which bacteria have, which is called recombinase A, which helps these holiday junctions form and stabilize, and it promotes this. Okay, so just think every time you see RecA promotes recombination. And this is actually a good time to talk about, we have enough time, a good time to talk about bacterial strains. When you look at bacterial strains, the most common strain notation you will see is RecA delta. Why? Why would we want to work with RecA delta?
So it will, like, it's not got the machinery already in place. If you want to be able to introduce that yourself.
Yeah, when we are doing standard cloning or normal cloning or heterologous protein expression, we're putting in a plasmid A. And one of the things we worry about is that plasmid A getting messed up, getting disrupted. And the most common way that that happens is if the cell uses RecA to recombine something in or out of the plasmid. Does that make sense? let's say your bacteria does not like to make protein X. It does not want to make protein X, but you as the researcher want to make protein X. If you try to force a bacteria to make protein X and it doesn't want to do that, it could use RecA to destroy that plasmid through recombination magic. It can recombine it with stuff on the chromosome. It could recombine it with some junk DNA that it found. Like, it can mess up your plasmid. So when we work with heterologous protein expression, or when we build plasmids, we want to ensure that those plasmids are not disrupted. So we actually work with RecA delta strains most often, because we just want to avoid the possibility that the cell could even recombine that. Okay, so this is looking at, does the recombination system, does it matter if, does it still work in delta RecA? It's testing, is RecA necessary, or can exo beta gamma do it independent in delta RecA systems? And RecA, if you have it, you do get higher efficiencies of recombination, and that makes sense. But most often we don't want to work in RecA strains. So it's nice that in the delta RecA strains, which is mostly what we're working with, you can still get it to work, although it's about a 10-fold less efficient. Okay, so that's what figure 5 is telling you. Okay, so we talked about RecA. Okay, let's just end with. So the idea is this can work. You can recombine. There's so many things you can do with this. You can recombine into plasmids. So you want to insert sequence X. You can do that into a plasmid by picking the homology regions that match the plasmid. You can recombine onto a chromosome if you want to do like a deletion or insert something by just picking homology regions that match on a plasmid region. There's also what's called a BAC, which stands for bacterial artificial chromosome. This is when sometimes we want to work with sequences that are like 100,000 base pairs. Okay. Oftentimes, if you're sequencing genomes of an organism, you will clone these things onto BACs. And you can only clone into BACs with recombination. It only works with recombination. You can't restriction enzyme clone into something that's 100,000 base pairs size, if that makes sense. Because your restriction enzyme is going to chew up your BAC just based on randomness of finding that sequence internally. Okay? Does that make sense? So if anything was unclear, it's because I will be completely honest, recombination is the system that I'm least familiar with. But I would say I've read this paper multiple times, and the paper is super informative, and it's super short, and it's super simple, and it's a famous paper. So read this paper, and you should understand, and watch the videos, okay? And everything that I expect you to know is on this. So I'll end with that. I was flustered this morning.